I'm ready to go. Thanks, yes, Dave gave a good introduction. I'm going to talk about DNA and eDNA approaches, and as you have already seen in Morgan's talk, we started using these approaches for quite a few different things. Frederica is going to talk after me about using fish faeces and using eDNA to detect CoTS in fish species. I'm going to talk about eDNA in general and about our larvae work. So, basically, I don't need to go too much of an introduction. Here we all know in the room that we're currently in the fourth CoTS outbreak since the 1960s. Most of these outbreaks have behaved quite predictably, as these data show here. They usually start somewhere in what we call the initiation box in the north, and we can all quibble a bit about whether that's closer to Cairns or to Lizard Island, and they progressively move down south. And practically by the time we get the outbreaks in this, well, the outbreak stopping at the southern end, we've got the next outbreak starting in the north. So it's probably even debatable if we're just having different waves of the same outbreak over the last 50 years. And in addition to that, we also find outbreaks in the Swains region, which may or may not always be the leftover from the previous outbreaks or an outbreak in itself. CoTS in Glenn Diaz's paper, for example, it has been shown that CoTS are very important in reef destruction. So at that point in the 2012 paper, CoTS were responsible for 42% of reef loss. That figure would be slightly more favorable for the CoTS in the moment, I assume, because so much coral has been lost to bleaching in the last 2 years. But in either case, it's a huge number, and you also, everybody in the room knows that we have competing or alternative or additive hypotheses. Why these CoTS are outbreaking? Is it a top-down control because the predators are lacking, as Dave Westcott had alluded to previously? Or is it a bottom-up control because the survivability of the CoTS larvae is increasing with more nutrients in the system and more food for the plankton? Or is it, which might filter through in the long term, a combination of these two factors, or rather local hydrodynamic factors? I'm talking about eDNA here, and that's going to be partially larval DNA, but also DNA detected from adults in the system. Why we started thinking about using genetic methods is that basically you need to be a really high-level expert to distinguish Crown-of-Thorns larvae in the plankton, even from sea cucumber larvae. That's doable, but even that, look like you see down here, they're very similar larvae types, specifically after they're fixed. So we originally started to develop these techniques to find more out about the larvae. What are the larvae doing? How are they dispersed? At what water depth are they? And how often do they appear? So here, in this talk, I'm going to talk about the larvae work, mainly in the context of the NEST project. So larvae, eDNA as tools for monitoring and early detection, but also finding out where larvae are in time and space, which is quite important for seeing where they travel and where they're coming from, but also it's quite important to know at what water depth they are, for example, in order to model their distribution. And then I'm going to briefly talk about developing techniques to detect juveniles and adults on the reef, using basically very similar genetic probes. So the concept here I have already preempted. eDNA in general is a term for environmental DNA that can be all sorts of DNA, which could be sediment bound, or it could be free in the water column, or it could be attached to particles in the water column, and that's not relevant. Or in some definitions also larvae DNA or microbes count as eDNA. So here we talk about larvae DNA, and I'm just going to give some examples on that. Basically, in collaboration with Jason Doyle at Ames, we have developed a qualitative method which we had already published 4 years ago, and can also quantify these things. Now, The second section of the talk is talking about eDNA, which is non-larval DNA, which is freely in the water column and which we take as an indication of a presence or absence of post-settlement Crown-of-Thorns starfish. Basically, we have initially tested these methods having a database collected of other sea stars as well on the barrier reef, and we've used GenBank to make sure that these probes are really specific, and had quite early on shown that these probes are specific. They're specific to Crown-of-Thorns, not to our local solaris, and they're detecting all 4 Crown-of-Thorns species. So that doesn't matter in the local context, but it matters because we could use these techniques in other regions of the world. So they're specific, and as I alluded to in our first paper on this, we had shown in 2014, we had conducted one long transect between Lizard Island practically, or the Cooktown region, and Townsville, and we showed that we can actually in December at this stage find larvae. So each of the red dots here is a positive detection, and at that stage we could not say how many larvae there were. However, we have further developed this technique into a technique called real-time PCR or quantitative PCR. We have used larvae available at Ames through our experiment we're running there to test how much DNA and how many copies you find. So going through a few loops, we could find out a way to actually quantify larvae in the sample. So currently we're in a position where we can quantify, not to the exact single one, but giving a range, a threefold range, and how many larvae we find in the samples. This was exemplified here with the same data set. Meanwhile, with the help of a lot of people, especially also Eric Fisher from Moore Reef and Quicksilver on Acanthaster Reef, and our own samples and AMPTO, we've got a database of over 2,000 samples which need to be analyzed. These are just the samples available in time and sorted by latitude. They're not, most of these are not analyzed yet, but so blue just means we've got them. It doesn't mean there's larvae or not larvae, just to show you how much material we got. And as one example, I'm going to show the time series which Eric and his team have collected on Moore Reef. So this is just an example of what you can do with these data. For here, we're looking at a time series just illustrating when we actually find CoTS in the water, CoTS larvae. So every green dot here is a positive detection, and it's not a surprise, but we can narrow down the spawning times a bit more. We find larvae in the plankton in practically December, January, and a little bit into February, and we start finding them pretty much exactly when the temperature goes above 28 degrees, which was previously predicted from studies in Japan. So it's a quite close match to that. Very briefly, coming to the adult side now. So for the adults, for the larvae, we need to take big plankton samples, whereas for adults we just collect 2 liters of water, but collect quite a few replicates. We quite early on figured out that the quantitative PCR method we used for the larvae is not sensitive enough, so we went another step using a technique called digital PCR, which can detect even smaller amounts of PCR products in water samples. So basically, we just collect samples, as indicated here by sample water samples, do our lab work, and then match these samples, map the samples onto the reef, and compare them with samples and transects we have taken on the reef. So for example, here we've got 11 reefs plotted. These are actual CoTS transects we have conducted at the same time. So these are CoTS counts on specific locations. Red again means no indication. So these are the Lizard Island area where we had an outbreak and where it's disappeared now. This is the outbreak front at the moment. And even by just mapping, so the next slide is going to be the actual eDNA concentrations on these reefs. So even without any statistics, we can see there's a very clear match wherever we've got CoTS on the reef, even in low densities, we can find DNA. So we now know that we can use this method to detect outbreaks and to detect early outbreaks. This just illustrates that there's even a reasonable correlation between the biomass and the eDNA concentrations for samples collected underwater or on the surface, which is quite important because surface samples are much easier to collect. So we can probably in the future even get surface samples to collect. And this is a correlation with the eDNA copy numbers, whereas this is just a correlation between the number of positive samples, which is also much easier to work out. So we might just be able to take more samples and might not even have to quantify all samples. So this is the work we've done so far. In the current nest phase, we're focusing on getting a better workflow worked out and an easier workflow. We're testing in collaboration with CSIRO some automated sampling devices here, which is actually happening on a field trip in August, coming up soon. So we're testing the first ones of these. Also, Jason has played a lot with a technique called dipsticks, simply, or lateral flow analysis, which works quite easy, and basically you can simplify the method to a point where you actually have, like for a pregnancy test, you just have positive or negatives and you can see what's coming out. We still have to develop the lab techniques and make them easier, but at the end it means operators, tourism operators could go out and do their own tests on the reef eventually. Then what I'm working a bit on, you've seen Mark Baird's talk this morning. We try with Barbara Robson at AIMS, we try to use the same model, the recon model, to find the best spot on the reef and the best time to collect and to place these samplers, because not every spot on the reef has the same water residence time, and obviously not all water flow conditions are the right conditions to collect these samplers. So what we've seen in the correlations, which were relatively good, was basically completely uncontrolled so far for location and for tides. But once we got the models worked out better, we can probably collect at specific tides and time and focus on that. So just in conclusion, I think we have the larval methods fully developed and worked out, so we can use these further to find out more about larvae ecology and find out a bit more about temporal resolution and look at other parameters which are needed for population modeling. This helps to understand causes of outbreaks, but it also helps us to provide management levers for things we can do to prevent these outbreaks. The post-settlement methods are kind of finalized, but they still have to be fine-tuned, and as I said, we have to find the right locations and times to sample, and we're conducting more tests. We can, basically this is here, especially in the field of automated samplers. And in summary, we think that both of these methods will help to help us to detect larvae, to detect CoTS outbreaks early, so that we can provide early intervention. Thanks, that's all.